Gayatri? Gayatri?
Good morning, sir.
Good morning, sir.
Am I audible? Gayatri, Lokeshwari, Sumaya, Janani. Ma, am I
Yes,
audible? உங்களுக்கு ஸ்லைட் தெரியுதாடா? பாலிமரஸ் செயின் ரியாக்ஷன் தெரியுதுங்களா? Anyone reply?
தெரியுது சார்.
ஓகே. ஸோ நேற்று நம்ம தியரியில், அதாவது உங்க நார்மல் கிளாஸ்ல பார்த்த விஷயம் தான். ஜஸ்ட் நான் ரிவைஸ் பண்ணிடுறேன். தென் அட்டனன்ஸ் எடுத்து நம்ம முடிச்சிடலாம். எனக்கு தேர்ட் ஒரு கிளாஸ் இருக்கு. ஸோ greetings of the day. PCR, otherwise known as polymerase chain reaction. The PCR technique first developed by Carey Mullis in the year of 1983. It utilizes tag DNA polymerase, an enzyme derived from the thermophilic bacterium Thermus aquaticus, which remains active at high temperature required for DNA denaturation. The basic principle behind the PCR technique is it used to amplify specific DNA sequences. It involves repeated cycles of denaturation, annealing, and extension. In the first step, the denaturation takes place at the temperature around 94 to 98 degrees Celsius. It separates double-stranded DNA into the single strands. In the second step, annealing takes place around the temperature of 50 to 65 degrees Celsius. It allows primers to bind to a complementary sequence on the target DNA. The third step is extension, where DNA polymerase enzyme synthesizes new DNA strand, adding nucleotides to the primers at the temperature around 72 degrees Celsius. What are the instrumentations are required for this thermal cycler? First one is we need the programmable machine known as thermal cycler, which perform temperature cycling for denaturation, annealing, and extension. Then we can discuss about what are the various reagents. First one is we need template DNA, the target DNA to be amplified. Second one is primers, the short oligonucleotides complementary to the target sequence. The third one is DNTPs, deoxynucleotide triphosphates. For example, ATPs, GTPs, UTPs, etc. TTPs, etc. Sorry, UTP won't become, it belongs to RNA. Building blocks for DNA synthesis. Tag polymerase, it is the heat stable enzyme for DNA replication. Buffer, maintain optimal conditions for the polymerase reaction. The detection system include for the quantitative PCR, it may be fluorescent dyes like SYBR green. In the other hand, probes like TagMan for sequence specific detection. If you list out the various types of PCR, the first one is conventional PCR. It is the standard method for DNA amplification. The second one is real-time PCR, otherwise known as qPCR. It quantifies DNA in real time using fluorescent markers. The third one is reverse transcription PCR, RT-PCR. We are aware about RT-PCR from the period of COVID-19 and converts RNA into the cDNA before amplification. Multiplex PCR amplifies multiple targets simultaneously with different primer sets. In the other hand, nested PCR uses two sets of primers to increase specificity. The sixth one is digital PCR. It provides absolute quantification of DNA molecules. The seventh one, hot start PCR. It reduces non-specific amplification by activating tag polymerase only at a high temperature. The eighth one is touchdown PCR. It gradually decreases annealing temperature to enhance specificity. The ninth one is asymmetric PCR. It amplifies one DNA strand more than the other, used for sequencing or probe generation. The final one is colony PCR. It amplifies DNA directly from the bacterial colonies without prior extraction procedure. Next, we can discuss about what are the various applications related to PCR. The first one is molecular diagnostics. It includes detection of genetic disease, example, cystic fibrosis or sickle cell anemia. In the other hand, identification of pathogens. Pathogens are nothing but disease-causing agent. You can be able to detect Mycobacterium tuberculosis, HIV also. The very good example nowadays, COVID-19 detection. Next important application is forensic science, DNA fingerprinting for crime scene investigation, paternity testing, who is the father of the boy or girl, if you go for the forensic science, genomics and research, the gene expression studies, sequencing and cloning of genes, analysis of mutation, it may be a genotyping, evolutionary biology, amplifying ancient DNA for phylogenetic studies, identification of extinct species DNA. In the other hand, food safety and agriculture, and detection of genetically modified organisms, GMOs, identifying foodborne pathogens. In the other hand, cancer research, detection of oncogenes or tumor suppressor gene mutations detection, monitoring minimal residual disease. The next application is environmental studies, detection of microbial communities in soil, water, or air sample, identifying bioengineered organisms. The final application is therapeutics, CRISPR-mediated genome editing validation, monitoring viral load, and antiviral-based treatment. If you see the pictorial representation of the PCR components, first we need the DNA sample, then we need the primer, then we need the nucleotides, DNTPs, CTP, GTP, ATP, TTP. It has to be put into the PCR tube along with the mixing buffer and a tag polymerase in the thermal cycler. The PCR cycle will take place like that. The PCR process, if you take the one cycle, first denaturing, the two DNA strands are separated. So double stranded DNA is converted into two different single stranded DNA. Each strand acts as a template for the another one. The annealing process takes place. Finally, extension. So one DNA is converted into two. So multiplication takes place. So 1 get 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32. This kind of multiplication takes place. These are all the information behind the polymerase chain reaction. Thank you. So remaining we will discuss little later. But now you can answer your presentation, answer your